So, Chapter 17 is all about taking the basics of what we learned about the immune system and the immune response and how we can use it for diagnostic purposes. So, we know antibodies are made and they target specific antigens. Well, because we know this, we can diagnose to see if a patient has antibodies. We can find out if they've been exposed to something or are currently being exposed to something. But we can also test for a specific antigen in the body using antibodies to detect that antigen. So it's all for diagnostic purposes. So in this chapter, we're going to go through how we get some of the background information when we examine them, how we can use their DNA, how we can use RNA to diagnose, how we've got some really fast rapid testing to give us really fast results because we don't like to wait for anything, how we can use those antibodies to help find and detect those antigens, how we can look for signs of infection, there's other diagnostic tools. So some of the methods we use to identify unknown microbes, that's what we're doing all the time with patients, is they come in, they're sick, we don't know what they have, so we've got to look at some different ways to diagnose what they have. One of the ways is using a phenotypic method. Is what do they look like? In a lab, under the microscope, is it a gram positive or gram negative? We can determine that by color. What do the bacteria look like on plates? What do those colonies look like? Are they big? Are they small? Do they have any certain colors or other distinguishing factors about them? It's like, what do they look like? If it's a bacteria, is it a coccus shape? Is it a bacillus? Is it a spiral shape? And I'm like, it's all these different, just what do they look like? Genotypic is using genes. This is where we're going to be using DNA or RNA to help diagnose. And then there's immunological ways. It's looking at blood, looking at other tissue fluids, and looking to see if there's an antibody-antigen reaction, meaning either the antibody is there because the patient made the antibody and they were reacting to an antigen, or we can look for an antigen by using antibodies in a lab to detect those antigens. So it's using an immune system, hence the immunological. And it's serology because we're using your blood serum to detect those antibodies. So the first type of genotypic method we're going to talk about is called the polymerase chain reaction, or PCR. It's an extremely fast method to detect for a specific, whether bacteria, whether virus, whether fungus, but we are looking specifically for that organism's DNA or RNA, depending on what they have. Now, we do have what's called a real-time PCR that can do it very, very quickly. And we can also, depending on the organism, do a reverse transcription PCR, meaning there are some organisms that have RNA, and we can do reverse transcription to make them make DNA, and then we can detect the DNA. That's what we're doing with COVID right now, is COVID has RNA, but we want to detect the DNA, so it does a reverse transcription. It's highly sensitive and highly specific. It's highly sensitive, meaning it can detect a very small amount of a microbe that's present. It's very specific because you're detecting the DNA specifically of that organism. So there shouldn't be any other conflicts or question on positive reactions. So we use PCR all the time to detect HIV, Lyme's disease, HPV, tuberculosis, hepatitis, and even, yes, COVID-19. Now, the immunological, this is again, we're using our immune system. We're looking at what's in the blood serum. So we call this process something known as serology. It's a diagnostic testing of the blood serum, and we're looking for some of those antibody-antigen reactions. We know antibodies with their arms and their hands at the top. We know those hands at the top, the variable region, is specific for some type of antigen, and we know its job is to grab on and hold on to that antigen. So we can look to see, well, that might cause, knowing that it does that, it might cause a precipitation. I'll show some pictures coming up. It might cause a precipitation type of reaction, which we can visually see. It may cause a color change in a lab, or it might release some type of radioactivity. Kind of depends on how we're testing them. So we can use serology to detect if there's an unknown antibody present, or using a known antigen. Or we can do a vice versa. If we want to know if a patient has an antibody, we can actually mix a patient's serum with a known antigen, and if they meet up together, we know they're both present. But vice versa, we can detect if if there is an antigen present in a patient's serum. We can add some antibody from a lab, mix it with the patient's serum, and if they react, well, we know the antigen is present. Now, using serology, it does have a high degree of specificity and sensitivity. Just like using DNA, we want a high degree of specificity and sensitivity. We want it to be very specific. We want to detect only one specific antibody or antigen and ignore everything else. But we also want it to be very sensitive, meaning if there's just a very small amount present of an antibody or an antigen, we want it detected. So this is just showing how we can take a patient's serum mixed with a known antigen, and if the patient has the antibody will react with that antigen, and it's usually going to form some type of visual kind of diagnostic tool. The one over here, this is how you can do antigens and antibodies mixed with blood typing, and we can visually see clumping happening. This is showing an example unknown serum on a card with 5 different known antigens and our positive control. And so those clumping indicates some of the precipitated antigens and a possible infection. So right now it looks like our patient has a Neisseria meningitis because that clumping happened, meaning they had the antibodies with that known antigen that was on there, and it caused that clumping to be visually used for a diagnostic tool. Again, this is just showing how we can take some antibodies of a known identity with some patient bacteria or patient sample, mix them together. This is how you can see those antibodies with their 2 arms can clump together something we can visually see and diagnose a patient. Again, sometimes you have to see the clumping underneath the microscope, and some we can actually make some of the clumping glow a little bit. So it makes it even easier to see and diagnose. Now, an agglutination test, because we're going to talk about both agglutination and precipitation, so I put this little picture on the bottom. They have a similar background, same kind of basic process that's happening. It's just what we're clumping or gluing together is different. So in a agglutination test, antibodies are going to cross-link. They're going to attach to whole cell antigens, meaning large, insoluble antigens, whole cells. So agglutination on here is those antibodies with their arms and hands at the end will attach, cross-link, whole cells, the whole bacteria, and they can form these very large, very visible, insoluble clumps. We can clump the bacteria together. We use this for blood typing. We can use this to diagnose various bacteria and even viral diseases. This is, again, is just showing some of that agglutination. You've got a whole cell, little structure on the outside, the epitope, and those antibodies are going to recognize that epitope, that little structure on this whole cell antigen, and it will clump it all together. Now, I'll go back to precipitation is that a soluble antigen is made insoluble by an antibody. So we're going to take these tiny soluble antigens, which means they are dissolved in a fluid, and we're going to use the antibodies to make them come out of the fluid. Now, it's called precipitation is because in a lab they would fall to the bottom of a tube. The word precipitation means to fall. So when you talk about precipitation, most people are thinking it's raining, it's snowing, it's sleeting, whatever else Wisconsin can have in a week. It's all these different forms of precipitation. Precipitation just means to fall or fall out of the sky. Well, for us, precipitation means we're going to take these tiny things that were completely dissolved in fluid, and we're going to clump them together and make them come out of solution. We're going to make them fall out of solution. And so we can take these tiny little molecules, clump them together. Now they're large enough, they will fall out of solution. Now, a titration. Some of you may have had a titer done, and some of you may have not, but you may have to have one done if you're going into the healthcare field. One of the most common times that a titer is done, usually for students that are taking this class, is for those that have had the chicken pox. like have had it, not the vaccine. The main reason for that is if you have had the chicken pox, generally you didn't go to the doctor for that. It's nothing they can do for it anyways, as, calamine lotion, anything to stop the itching. But that also means that on your record, your medical record, there is no actual documentation that you've had chickenpox. So maybe it wasn't chickenpox that you had. So they want to know if you've been exposed to it in the past. And they want to know how strong is your immune system at recognizing this particular virus in the future. So if you did work with a patient that had the virus, are you safe? Do you have that immunological memory? So they're going to do a titer. And so again, you may have had this done. Now, what they end up doing is they're going to take some patient serum and they're going to add it to these but they're going to increase the dilution. So they'll just take some straight up patient serum and mix it in here with an antigen. And this antigen could be for the actual virus itself. So we can say there's some chickenpox virus in here. And we want to know if you have had the chickenpox, you should have antibodies in your body. Now, those antibodies will mix with the virus that's here, and you're going to see clumping happening. However, with the titration, we want to see how many antibodies do you have. So we'll start to dilute that patient serum. And we want to know how far can we dilute out? How far can we dilute those antibodies that we still see a positive reaction? And in this example, I'm like, we diluted that patient serum all the way out 1,000 times diluted. And there was still enough antibodies to cause a positive reaction. So this will tell you that, one, like you've got enough antibodies that if you were exposed to this particular virus in the future, you have a strong enough immune system, you wouldn't get the virus. Otherwise, if your titer, now they call the most dilute that your serum can be and still show a positive reaction, they call it as a titer. And you would like a high titer. It means you have a lot, that's a big dilution, a lot of antibodies. Now, if your titer showed up as very low, maybe even non-existent, then they might say, okay, well, you need a vaccine. Or for a lot of individuals, you need a booster shot. They might do this for tetanus. If you're like, it's been a long time since you've had a tetanus shot, they might want to see, how is your immune system reaction? If it's your titer is way back here, then you might need a booster shot. Now, they can also use titers on patients, not for just looking at, if they have antibodies in their immune system. It can determine, we know what antigen we're adding in here. So if we see any clumping, that can help us determine that, yes, the patient has antibodies. But we can also look to see how is their immune system handling a particular infection. So if there's an infection present, the body should be increasing antibodies every day. As their immune system is recognizing it, we're waking those B cells up, they're going to turn into plasma cell, and they're going to start churning out tons and tons of antibodies. And so they can do a titer. They usually will do a titer every day, every other day, and they're going to look to see, does that titer start to increase? If it does, that means the patient has an infection because they are recognizing it, it is mounting an immune response, and we're seeing that increasing level of the antibodies because you're going to have an increase in titer. So we can use it to diagnose them based on if you see a positive reaction or not, so you know what it is, but you can also say that, yeah, there is an infection and their immune system is responding. If 2 weeks go by and their immune system is not responding, they might have some type of immunodeficiency disorder, and you might need to treat them with a little different type of a different type of drug. You might need to use a bactericidal versus a bacteriostatic type of drug. Now, another type of serology is the Western blot. It's looking for proteins that the actual organism has itself. So in a Western blot, we can actually separate out proteins by size. So we can take an organism and we can break apart that organism and we can separate all of its proteins out by size. It's a highly specific test. It's highly sensitive. And we're going to look for various types of antibody binding patterns. Again, the antibodies are going to bind to the different proteins, and we're going to make it visual so that we can see where those antibodies attach to those different proteins. Now, a lot of times for the Western blot, a Western blot isn't done all the time. It's mostly because it's kind of putsy, which means it's kind of expensive. And if we've got other faster, easier, cheaper methods for detecting, we usually do it. But we do usually use the Western blot as like a second test for HIV. It's kind of the confirmation test for HIV as well as for Lyme's disease. So this is showing how we can separate them out. I'm going to come back to this because I like this one for describing what's happening. Is we can break apart the microbe. Then we're going to separate it out by size. We do that on this gel, is we subject it to electricity. The smaller proteins, we put them all in over here. This is like six different patients. We put the patient's organism in there. That organism has been broken down. We shoot it with some electricity, and the smaller proteins travel farthest in the gel. Mostly because they're tiny and they can wiggle their way through the gel really, really easy. Really big, fat proteins can't move through that gel very quickly, so they're going to hang out way back here. So we're going to separate these proteins out by size. Now, it's called a blot because we have to get all of these separated out proteins from a gel onto a paper that we can mix with antibodies. And so it's blot because we take, they say an absorbent paper. Usually it's your kitchen sponge. And we've got the gel right here, but right here is where we want those proteins to be on. We want it to be on this little membrane type paper. And then it has filter paper. Paper towels work fine. Well, if you take a wet sponge and dry paper and you push down, if you took some dry paper towel and pushed down on a wet sponge, what's going to happen to the paper? Well, the water is going to go from the sponge up onto that dry paper. Same basic idea, except when the water goes from the sponge up onto that dry paper, when you squish, that's the blot, when you squish down, is that as the water goes up, it actually picks up the proteins with it, deposits it on the paper, and then it continues onto the filter paper or your paper towel. So it's one quick, easy way is we're using water and movement of water to move the proteins from the gel onto the paper. So we've got all of our proteins on that special membrane paper. We add colorful antibodies, and wherever the antibodies stick, meaning they recognize those specific protein fragments, wherever they stick, we see a colorful line. Now, we always do this with a positive control to make sure everything worked, and we do it with a negative control to make sure we don't have any contamination. And we have our 6 patients right here. And so generally the rule is for Lyme's disease, because there are lots of different proteins and lots of different things, when we separate them out, the CDC recommends that a patient has to show that they have 5 out of the 10 main Lyme's disease proteins. So on here, on these 6 patients, whoa, which ones have Lyme's disease? Well, patient 3, for sure. Like, they are matching up with a lot of those proteins found in that positive control. 5, yep, definitely has Lyme's disease. And then I would say even 6. Some of those proteins are small, but if they show up at all, it means they are there. So again, here's another one. Yes, CDC requires 5 out of the 10. We've got the first column is our negative control. 8th column is our positive control. Out of our patients, 2, 3, 4, 5, 6, and 7. Who has Lyme's disease? Well, we're looking for at least 5 bands, definitely 3, as well as 2. So we use this. It's very sensitive. It's very specific, looking only for this particular organism. So again, we can use this. We use it to test for HIV. We use it to test for Lyme's disease. They're a couple of the most common ways. These are the glycoproteins, the proteins that we have separated out by size. And you can see how some of them match up, but they don't always in all patients. Now, another way we can easily see if these antibodies are there or not is we can label them. Is we can actually put like almost little tiny flags on the antibodies in our lab. And one of the things we can add onto the antibody is a fluorescent dye. So then wherever that antibody sticks, when it finds its antigen, we can easily see that fluorescing very, very well under the microscope. I'm like, sometimes we don't even need the microscope because it fluoresces so brightly. Now, a direct test is that we're going to have some type of unknown test specimen or antigen. We're going to put it on the slide. We're going to expose it to that fluorescent tagged antibody, and we're going to look to see if that fluorescent tagged antibody sticks to the antigen that was added. We are testing for the antigen. So a direct test is testing for the antigen. We want to know, does that patient have that bacteria present? Is that virus present? Is that fungus present? Is that parasite present? We're looking for the antigen. And we do that by adding some fluorescent dyed antibodies. If they stick, that's because the antigen is present, and we can easily see that. Now, an indirect test, we're going to reverse it, as we want to know, is the antibody present? Does the patient have antibodies against a particular organism? Well, we can still add fluorescent dye to antibodies, and antibodies can stick to the constant region of other antibodies. So antibodies can stick to antibodies. Now, if a patient has antibodies, the fluorescent colored ones will attach to them, and underneath the microscope, you're going to see things fluoresce. It can tell us right away that the patient has a very specific antigen if it's the direct test, or we can see if a patient has a specific antibody to a specific antigen. We can say, does the patient have the antibody for the chickenpox virus? We can see if they have the antibody for a particular bacteria, which means they've been exposed to it. They've got the antibodies in their body system. Now, immunoassays. These are extremely sensitive, and they're used to trace or to detect trace antigens or antibodies. So if we don't expect to see a large quantity of an antigen or even a large quantity of antibody, we can use some of these immunoassays to look for very, very small, minute antigens and antibodies. One example, it's a radio immunoassay. It's the antigens or the antibodies, depending on what we're using in the lab to determine if a patient has antigens or antibodies. But we can actually take antigens and antibodies and we can link them. We can label them in the lab, not with a fluorescent dye, but instead with radioactive isotopes. And so we can look to see if they stick to the patient sample. We're going to see that radioactivity present, and any radioactivity present, you can detect very small amounts of radioactivity, which allows us to detect very small amount of antigens and antibodies. Another way we can do an immunoassay, instead of labeling the antibodies with a dye or radioactive isotopes, we can label them or add an enzyme to them. Now, enzymes work on different substrates to create different products, and we know that. So we can actually add an enzyme that we know is going to give us a color change. And so we know if the enzyme's present, it's because the antibody's present. It's stuck to whatever we are hoping it will stick to, whether it's an antigen or an antibody, but it's going to stick to what we're testing for, and we'll see a color change. Now, the enzyme-linked immunosorbent assay, it's long, so we call it an ELISA. We can use it to detect the antigens or antibodies, so direct tests as well as indirect tests. Now, an indirect ELISA is we are looking for a patient antibody. So we want to know, does a patient have an antibody? I'm going to look at a different picture. So this is, we're going to have the indirect test. And so we can add an antigen. This all happens in these little plastic plates. We can add an antigen. So we'll pretend this antigen, this red antigen is, I don't know, staph. No, I don't like it. E. coli. That would look more like a little pink oval. So we're going to say that this is E. coli. We want to know, does a patient have antibodies to this particular E. coli? So we'll add the E. coli. We're going to add the antigen to the plate, and it will stick. They're made to be sticky. We cover everything else up. We add that patient serum. If that patient has antibodies to this particular antigen, and it does in our example, it will stick. We rinse off any excess. Then we add our enzyme-linked antibody. So this particular, we added an enzyme to it in a lab, and this antibody can recognize the constant region of other antibodies. So they stick. Now, we add a substrate that this enzyme works with, and that enzyme takes the substrate, produces a color change, and we can see exactly which patients have the antibody. That's an indirect test, is we're looking for the antibody. We can do the whole thing and reverse it and do as a direct test. If we want to know, does the patient have the antigen? Does the patient have E. coli? So this is kind of the quick version of it, is that you can add antibodies that are specific for E. coli to the plate. They'll stick. Add the patient serum. Our patient has E. coli, or whatever it is we're testing. So that antigen is going to stick to those antibodies that are already in the plate. Then we add our enzyme-linked antibody, which recognizes the antigen we're testing for. We add the substrate, we get the color change, and again, we can visually see who has it. Now, a little note, you might be able to notice that some of these blues are different colors. All the blues are positive reactions, but some are darker blue, some are lighter blue. Well, any color change means that enzyme is present, means whatever you're detecting, antigen or antibody is present, but it can help us quantify a little bit as well. The more antibodies that stuck, the more antigens that stuck, the darker blue reaction. So this patient's going to have more antigens or antibodies, depending what you're testing for, compared to this patient. Either is a positive reaction, but it can help us quantify that as well. So this is showing that we're detecting, these are the ones out of your book. I found this one somewhere else. I liked both. This one's detecting that it's a direct test. You're looking for the antigen. So we have that enzyme-linked antibody attached to the patient antigen. It's there. We get a color change. And I'm like, again, this one is showing there's 96 tests. And again, it's one plate that you can actually test 96 different people for. So you can do a lot. It's like mass testing. Anyone on here that shows up as yellow, this one's yellow versus blue, and I'm like, they would show up as a positive reaction that that patient has a particular antibody or it has a particular antigen. Now, we don't like to wait for tests because sometimes they want to wait till they have 96 different people to test for something. Some things we have to grow up in a lab. We got to wait 24 hours, then run another test, another 24 hours. It can take a long time for some of these tests. I mean, once they run them, we're getting faster and faster with them, but they can sometimes take a while. And generally, when you go to the doctor, you'd like to be able to leave the doctor's office with a diagnosis. And so we are developing what are called point-of-care tests. They're simple immunoassays. So they're going to take some type of patient serum, and they're going to give a quick diagnosis. We don't have point-of-care testing for everything, but we do have it for some. And so some, you just take a little bit of patient sample, sometimes just a blood sample, sometimes it's a sputum sample. You can put it on a filter paper. It's called an immunofiltration assay. It's on a piece of filter paper. There's already antibodies, labeled antibodies on the filter paper. And if you have the antigen, it will mix with it and show a color change to give you that quick, right away, within minutes result. In immunochromatography assay, similar. You have labeled antibodies on paper, but instead of putting them directly on those antibodies, we let a patient sample kind of travel up a strip of paper until it meets up with those labeled antibodies. Then we get the color change. Now, some examples of an immunochromatography assay. A pregnancy test is an example of immunochromatography assay. So when you pee on the stick, that urine has to travel up the stick until it meets up with a labeled antibody. And that labeled antibody is specific for a hormone, the pregnancy hormone, and the HCG. And if that HCG is present in the urine, it reacts with the antibody that's on the paper, and you get a color change. Now, there's a reason why it's always one line and two lines. One line always meaning not pregnant, two lines meaning pregnant. The one line will show up. It will indicate that you just had enough urine to make it that far, so that the test is accurate, that you peed enough. The other line is the line that's detecting the hormone that's determining pregnancy or not. So there was just another one. We also have it that we can do the same thing for mono. So if you go to the doctor's office and go in for a mono test, they can give you those quick results. Same thing with the strep test. So like if you've ever had strep throat, you go in, they take a throat swab, they leave, you sit there for a while, and they come back and they say, yes, our tests indicate you have strep throat or you don't. Well, they did one of these things. I'm like, it's generally not in this little tube, but same basic idea is they take that throat swab and they break apart the antigens that are there. They run it up a strip, and if it encounters the labeled antibodies for the strep antigen, it shows up as a color change. It says, yes, you have strep throat. So I'm like, different really quick ways we like to get test results. Again, we don't have this for all of them. We're getting there, though, because people like fast results. We all do. Otherwise, if you have any questions on this PowerPoint, again, please send me an email. I'm happy to answer any questions.